Good afternoon, everyone. My name is Dr. Keshav Murthy. I am professor and HOD of Biochemistry department. Today, I am taking class about gluconeogenesis. Okay, this is the continuation from the next chapter. Gluconeogenesis is the reversal of glycolysis. Okay, Prashant sir talked about glycolysis. This is the continuation part. Okay, gluconeogenesis, as the name itself suggests, gluconeogenesis. Gluco means glucose, neo means new, genesis means synthesis. Synthesis of new glucose from non-carbohydrate precursors is called gluconeogenesis. Okay. So we'll go to the next slide. So what are the precursor? What are the substrates for gluconeogenesis? One is amino acids, alanine, glycerol, lactate, propionate, and carbon sources. These are nothing but fats, breakdown of lipids and proteins. Okay, breakdown product of proteins are amino acids, especially alanine. Okay, alanine is the most common amino acid which gives rise to the glucose, and glycerol. Glycerol is the breakdown product of triglycerides, and propionate. Propionate is the breakdown product of fatty acids, odd chain fatty acids. Also, we have lactate, which is the product of anaerobic glycolysis. All these will become precursors for gluconeogenesis. So as you see, this is nothing but gluconeogenesis is nothing but the reversal of glycolysis. Okay. So in glycolysis, we have three irreversible steps. One is catalyzed by hexokinase step, and another one is phosphofructokinase and pyruvate kinase. So these three enzymes are circumvented by four key enzymes. Hexokinase is circumvented by glucose 6-phosphatase enzyme. Phosphofructokinase is circumvented by fructose 1,6-bisphosphatase, and pyruvate kinase. First, pyruvate will be converted to oxaloacetate in the mitochondria. Okay, the enzyme is pyruvate carboxylase. Okay, these carboxylation reactions require three key ABC. These are called ABC reactions, which require, that is, A for ATP, B for biotin, and C for carbon dioxide. Okay, so pyruvate will be converted to oxaloacetate, but oxaloacetate cannot go back to the cytoplasm. So where does this gluconeogenesis occurs? It occurs mainly in the cytoplasm and mitochondria, both in the cytoplasm and mitochondria. This pyruvate will enter the mitochondria and it forms oxaloacetate, but oxaloacetate cannot go back to the cytoplasm. So oxaloacetate will be first converted to malate by malate dehydrogenase enzyme. The malate can go out of the mitochondria and into the cytoplasm. There, malate will be reconverted back to the oxaloacetate by phosphoenol pyruvate carboxykinase. Okay, this enzyme requires GTP, and one carbon dioxide will be puffed out. So this phosphoenol pyruvate which is formed will go reversal of glycolysis will lead to the synthesis of glucose. Okay, this is about gluconeogenesis. As I told you, this is nothing but the reversal of glycolysis and Krebs cycle. Okay, Krebs cycle will where so many intermediates enter. And so what are the precursors? Now we will see about the four precursors or substrate which will lead to glucose. First is glycerol. Okay, glycerol is the breakdown product of triglycerides. Okay, triglycerides are made up of nothing but the glycerol backbone. We have three fatty acid molecules. Okay, from that breakdown product is glycerol. So what will happen to this glycerol? Glycerol will be converted to dihydroxyacetone phosphate by glycerol 3-phosphate. Okay, so this dihydroxyacetone phosphate will be converted to glyceraldehyde 3-phosphate. Okay, so then it will run in the opposite direction, the pathway to leading to the formation of glucose. Then we have, as I told you, lactate which is the product of anaerobic glycolysis. So this lactate can be converted to pyruvate by lactate dehydrogenase enzyme. Then pyruvate will run in the reversal of glycolysis leading to glucose. Okay, Now we have seen how glycerol will lead to the synthesis of glucose and lactate will lead to the synthesis of glucose. Then we have propionyl coenzyme A. This is nothing but the breakdown product of odd chain fatty acids. Breakdown propionyl coenzyme A or propionate is the breakdown product of odd chain fatty acids. It will be converted to succinyl coenzyme A. First it will be converted to succinyl coenzyme A. Then succinyl coenzyme A will form oxaloacetate in the Krebs cycle. Okay, it's the intermediate of Krebs cycle. It will form oxaloacetate. Then it will lead to the formation of phosphoenol pyruvate, and reversal of glycolysis will lead to the glucose synthesis. Okay. Then we have glucogenic amino acids. Glucogenic amino acids. All amino acids will lead to the formation of glucose. All amino acids except leucine. Leucine is a purely ketogenic amino acids. Except leucine, all others are glucogenic. That is, all other amino acids can give rise to the glucose. Okay, so now we will see which all amino acids will lead to what all intermediates. So glycine, alanine, serine and cysteine will enter here. It will lead to the formation of pyruvate. Then the reversal of glycolysis will give rise to the formation of glucose. Okay. Then the amino acids glutamine, glutamate, arginine, histidine and proline will give rise to the formation of alpha ketoglutarate. Then alpha ketoglutarate will form oxaloacetate and it will lead to the reversal, will lead to the synthesis of glucose. Then isoleucine, methionine and valine will give rise to succinyl coenzyme A. Will form succinyl coenzyme A, and again these intermediates will run in the opposite direction to form glucose. Then propionyl coenzyme A, phenylalanine, tyrosine, asparagine and aspartate will form fumarate, and asparagine and aspartate can give rise to oxaloacetate. So thus all these glucogenic amino acids except leucine, all other glucogenic amino acids can give rise to synthesis of glucose. Okay. So what all we have to remember? This is nothing but the reversal of glycolysis and TCA cycle, except we have to remember 4 key enzymes. 4 key enzymes. Which are the 4 key enzymes? Glucose 6-phosphatase, which helps in the conversion of glucose 6-phosphate to glucose, and fructose 1,6-bisphosphatase, which will remove the phosphate from fructose 1,6-bisphosphate to form fructose 6-phosphate, and pyruvate kinase, pyruvate carboxylase, which will convert pyruvate to oxaloacetate, and phosphoenol pyruvate carboxykinase, which is called PepCK. These 4 enzymes are called the key enzymes of gluconeogenesis. Okay, you have to remember these 4 enzymes: glucose 6-phosphatase, fructose 1,6-bisphosphate, PepCK and pyruvate carboxylase. These are the 4 key enzymes of gluconeogenesis, and you have to remember where all each substrate enter. Okay, glycerol will form dihydroxyacetone phosphate, which will lead to the formation of glyceraldehyde 3-phosphate, and lactate will give rise to pyruvate by lactate dehydrogenase enzyme, and propionyl coenzyme A will form succinyl coenzyme A, which is the intermediate of TCA cycle, and all the glucogenic amino acids except leucine, that is alanine, serine, cysteine will form pyruvate, phenylalanine, tyrosine, aspartate, asparagine will form fumarate, asparagine and aspartate will form oxaloacetate, isoleucine, methionine and valine will form succinyl coenzyme A, and glutamine and glutamate, arginine, histidine and proline will form alpha ketoglutarate. Okay, this is about gluconeogenesis. Okay, the same thing has been explained in another diagram. 4 key enzymes. You can see the 4 key enzymes: glucose 6-phosphatase, fructose 1,6-bisphosphate, phosphoenol pyruvate carboxykinase, and pyruvate carboxylase. Okay, it has been shown here. Okay. Sources of, okay, this we have already seen. I will not go into detail here. Conversion of oxaloacetate to PepCK via the same thing. The same has been explained in detail. Oxaloacetate will be converted to phosphoenol pyruvate by PepCK, phosphoenol pyruvate carboxykinase. It requires a GTP and a carbon dioxide will be puffed out. Okay. So how lactose will be converted, lactate will be converted to glucose. I have already explained in the diagram, same diagram. Lactate will be converted to pyruvate by lactate dehydrogenase enzyme, and pyruvate is converted to oxaloacetate by pyruvate carboxylase enzyme, and oxaloacetate will be converted to malate. Malate will go out of the mitochondria into the cytoplasm, where it will give rise to again to oxaloacetate by, and oxaloacetate will give rise to phosphoenol pyruvate by PepCK enzyme. Okay, the same thing. Okay, how glycerol will give rise to glucose? Glycerol is nothing but it is obtained by the breakdown of lipids, triglycerides. Okay, this glycerol will be converted first to dihydroxyacetone phosphate, which will give rise to glyceraldehyde 3-phosphate. Okay, then by the reversal of glycolysis you will get glucose. Propionic acid, each one they have explained. What all the reactions I explained in the diagram, the same thing, each step they have explained in detail. Propionic acid, I told you, propionic acid will give rise to propionyl coenzyme A, and propionyl coenzyme A will first form methyl malonyl coenzyme A, and it will give rise to succinyl coenzyme A. It is the intermediate of TCA cycle, and it will lead to glucose formation. Then, as I already told you, these are all the amino acids which can give rise to glucose. That's why they are called glucogenic amino acids. As I already told you, except leucine, leucine is a ketogenic amino acid. Except leucine, all other amino acids will give rise to glucose. So they have mentioned all the amino acids. Pyruvate, alanine, pyruvate. Okay, these amino acids will give rise to pyruvate. Alanine, cysteine, glycine, serine, threonine, tryptophan will give rise to pyruvate. Arginine, glutamate, histidine, proline will give rise to alpha ketoglutarate. Aspartate, asparagine will give rise to oxaloacetate, and phenylalanine, tyrosine will form fumarate. Isoleucine, methionine, threonine and valine will give rise to succinyl coenzyme A. This I have already explained in the diagram. Okay, so the one key thing you can remember is most of these amino acids will give rise to alanine. Okay, alanine, then this alanine will increase. Okay, the alanine content will increase 4 to 5 hours after the intake of food. So the difference, the key enzymes, they have differentiated. So in glycolysis, whereas exokinase, these are the irreversible, these are the 3 enzymes which catalyzes the irreversible steps. So the exokinase will be circumvented by glucose 6-phosphatase. Phosphofructokinase will be circumvented by fructose 1,6-bisphosphatase. Pyruvate kinase will be circumvented by pyruvate carboxylase and phosphoenol pyruvate carboxykinase. Since there are 3 irreversible steps, these will be circumvented by 4 key enzymes. Okay. Now we will come to the reciprocal regulation, how these glycolysis and gluconeogenesis are regulated. So as I told you, both glycolysis and gluconeogenesis are almost opposite pathways except for the irreversible steps. Okay. So these regulation is also reciprocal. Reciprocal means whereas what all substrates and what all inducers and what all the hormones they will induce this pathway will be inhibiting the other pathway. That is why it is called reciprocal regulation. Now we will see here. So in glycolysis, the inhibitors and inducers are fructose 2,6-bisphosphatase and AMP. These are the activators, allosteric activators. Whereas here, in gluconeogenesis, citrate is the allosteric activator for fructose 1,6-bisphosphatase. And here in glycolysis, ATP citrate and H plus ion. These are allosteric inhibitors. Whereas here AMP and fructose 2,6 bisphosphatase are the allosteric inhibitors. See here, compare here. What the activators have become inhibitors here. So the both activators, allosteric activators, have become inhibitors for this enzyme. That's why it is called reciprocal regulation. So next is pyruvate kinase. Okay, pyruvate kinase activator is fructose 1,6 bisphosphatase and inhibitors are ATP and alanine. Whereas here ADP is the phosphoenol pyruvate carboxy kinase. ADP is the allosteric inhibitor, and for pyruvate carboxylase acetyl coenzyme is the activator and ADP is the allosteric inhibitor. Gluconeogenesis. So coming to the summary, I will summarize in few words. Summary is gluconeogenesis is nothing but synthesis of new glucose from non-carbohydrate precursors. What are the non-carbohydrate precursors? These are glycerol and propionyl coenzyme A, lactate, pyruvate and glucogenic amino acids. These are the substrates. And we have to remember four key enzymes of gluconeogenesis which circumvent the irreversible enzymes of glycolysis. Which are the four key enzymes? They are pyruvate carboxylase, PepsiK, that is phosphoenol pyruvate carboxy kinase, and fructose 1,6 bisphosphatase and glucose 6 phosphatase. And the reciprocal regulation, you have seen the reciprocal regulation of gluconeogenesis and glycolysis, and we require 6 ATP. 6 ATP is required for the gluconeogenesis. Okay, that's about today's class. Thank you.